thesis defense from Mateo Dávila-Jativa, which is entitled "How Do Opposing Pressures of Sexual and Natural Selection in Continental and Insular Settings Shape the Evolution of Coloration in a Tropical Gecko?" So Mateo, you have 30 minutes to present your thesis. Then we will have questions from Devi Stuart, who is from the University of Melbourne and is participating as the external evaluator of your thesis. And we thank Devi for being here and for evaluating your thesis. Then we will have questions from Andrew Crawford, a professor in our university, who will be the internal evaluator. And then we will go on to a short time for public questions, which will work from the chat in YouTube. We will not have live questions, live voice questions. And then we will move on to comments from your director. So you have 30 minutes from now.
All right.
Thank you.
Thank you. And thanks to everyone for being here. So I'm actually here to tell you a story of how this whole project came to be. So it all started walking down a really long road with a really good friend in San Cristobal Island in the Galapagos archipelago, where this friend and I, we had been working with this little gecko you guys see on the screen right now for a while. And during this walk, we started wondering what island life had done, or how island life had affected the evolution of this little gecko. Because as we know, natural selection selects for traits that are advantageous for an individual in its environment. However, sexual selection tends to favor traits that might increase chances of reproduction, but can be really costly or even detrimental to the survival of the individual in its environment. So these two opposing pressures working on the evolution of conspicuousness, of how colorful, how easy to spot this gecko was the thing that got us thinking and talking. And so I went into the literature and started looking at stuff, and I found this little quote from Endler's classic work on guppies from the 1980s, which states, "What is conspicuous to a predator will also be conspicuous to a female." And he actually tested for this. You know, there are these two huge evolutionary pressures working on a single set of traits. And so he tested for this, working with male guppies. And what he did was place these guppies under different predation levels. You can see on the x-axis on this graph, from left to right, they go from lower to higher predation levels. And as you can see, the length of the spots shown on the y-axis on this graph, the size and the amount of the spots, of the colorful spots on the male geckos, will decrease when under higher predation pressure. But upon release from this pressure, the spots in the populations will grow again. And these spots are a means of sexual signaling in guppies, which have a very strong sexual selection as well. This same pattern is actually described in other species, such as the dragon lizard, where in this species, colors that are more exposed to potential predators, specifically avian predators, such as the head or the lateral line, the dorsal line in this gecko, tend to resemble the color of the environment or the substrate where they lay, a lot more than colors that are more hidden, such as the yellow throat or the orange patterns you see on the sides. And you can see this on the reflect on its reflectance curves. I don't know if you can see this, but there's a gray line that goes through here, or a gray curve that shows the average color of the environment. And as you can see, the more exposed color, or the more exposed body parts appear to resemble the environment a lot more than these hidden body parts, such as I said, the throat or the sides. And as I mentioned, we were working in this island at the time with this gecko, and we knew, or it is a well-developed idea, that predation pressure in islands tends to be reduced due to a lower diversity of predators in general. So this predation pressure appears to be relaxed in archipelagos or in islands. And specifically in this archipelago, we have some really extreme examples of phenotypic changes or evolutionary changes on different individuals believed to have occurred due to this release on predation pressure, such as island gigantism in tortoises, or even the loss of flight on the cormorants. So we had this gecko living in the island as an introduced species, while at the same time populations from the same species were evolving and living in literally the middle of the tropics, right? The Ecuadorian lowlands, which are right in the middle of the tropics, where predation pressure is expected to be very high. So I guess I'm getting a little bit ahead of myself, and I should introduce to you the model for our study, right? So this is Gonatodes caudiscutatus. It's a dichromatic gecko. As you can see, the male geckos shown in the graph above the females and in the right have a very strong or very conspicuous, very noticeable yellow patterns in the head and blue dots on the rest of its body, while the females are a lot more inconspicuous. They're a lot better at hiding in their environment. This gecko is mostly diurnal, it's mostly terrestrial, and what makes it a very good model to check for what we had been talking about, it's its distribution. This guy occurs all over the western Cordillera slopes in Ecuador. And the thing is that in the Cordillera slopes, in the lowlands and the western Cordillera slopes of Ecuador, and to the north, it lives in really wet, rainy forests. But in the south, it lives in really dry forests. So the environments change a lot. And it also lives along a very wide altitudinal gradient. It goes up to 700 meters in the Cordillera. And as I mentioned before, it's been introduced in the Galapagos Islands. Specifically, we know it's been in San Cristobal Island. The first reports come from back in the late 1800s. And this is a relatively small island, almost 1,000 kilometers off of the coast of continental Ecuador. And it has a really interesting climate, or a very interesting ecosystems occur there. In the lowlands, this is a very small island, as I mentioned, but the lowlands are very dry, very arid, while the highlands are a lot more humid. And in such a small area, this change is quite impressive. So all of this context takes us to our research question. Is there a relationship between predation pressure and conspicuousness on continental and insular populations of Gonatodes caudiscutatus? And what we expected to see was that, sorry about that, in the continent, these two evolutionary pressures, right? Sexual selection shown with the yellow arrow, and predation pressure shown with the gray arrow in this graph, will have most geckos, like the number of geckos is shown in the y-axis, somewhere in the conspicuousness axis, but in a rather narrow distribution, right? Or a rather narrow variance. While in insular populations, we expect a reduced predation pressure, shown with a smaller gray arrow, and so this will give us more colorful geckos, right? The reduction of this pressure should allow for them to become a lot more colorful. And so with all this baggage, went into the field to actually check for this. And so we visited 37 different continental sites in 3 different types of ecosystems, in the northern wet Chocó lands, in the western Cordillera slopes, and in these very dry lowland forests in the south. And we also visited 15 insular sites, both in the lowlands and the highlands of San Cristobal. And the first thing we needed to actually check for was if there was a different predation pressure. So in order to do this, we built 800 paraffin models to look like the male geckos, the male gonatodes. And we took them to the field and placed them in 4 different locations along transects to check for predation markings. And what we found so far, no big surprises here, was that in general, continental geckos were under a higher predation probability, or had a higher number of predation or of attacks than insular ones. The continental geckos are shown here in blue, and the continental ones in orange. Sorry, the other way around. The continental geckos are shown in blue, while the insular ones are shown in orange. So the next thing we needed to do was actually to check for conspicuousness, and so we took it out into the field again, and we collected almost a little over 130 individuals, almost half of them in the Galapagos and almost half of them in the continent, and we actually checked for measured colors from them. We wanted to know in terms of colorfulness how conspicuous they were. So we took spectrophotometry curves from different body parts for males and females. On females, we measured the head, the dorsal line, and the sides. And on males, we measured all the different colors we could find in the body. So the very colorful yellow throat, the black and yellow head patterns, the dorsal line, these very bright blue spots on the sides, and the dark sides that they showed. Once we had these measurements, these reflectance curves, we used a tetrahedral color space model to turn them into points, or into points in a color space. So what this model does is actually pretty cool. It incorporates information from the different types of cones in the eye system of potential predators, in this case birds, and each vertex of the polygon represents the maximum stimulation for each of the 4 cones. So once you have points in a space, you can do some really cool stuff with them. So the first thing we did was measure colorfulness for each individual. So the way we did this, or the approach we took for this, was to measure color volume. So in order to do this, again, you take the smallest convex polygon that will encapsulate all color points from a single individual, and then you measure the volume that this convex polygon has, and this will give you the color volume for that individual, and this is a colorfulness measurement. Once you have that information, you can do some other cool stuff, which is compare colorfulness between individuals. And so in order to understand the color variation in our geckos, we did this for 2 males from each different type of ecosystem we sampled at. And what we get is these spaceship-looking shapes that are actually the 2 color volumes overlapping with one another, and you can measure how similar they are by how much they overlap between the 2. And what we found was actually pretty exciting. So we found a very big variation between males of these geckos along its distribution, both between and within populations. It was pretty surprising that in the same population you could find geckos with very dark backgrounds and very bright heads, such as the one shown down here in the bucket of geckos, and then geckos a lot more a lot less contrasting with themselves. We found an average variation within populations of 55%, so that they're pretty different between each other in the same population, but a way higher variation average between populations. So geckos that came from different populations were more different from one another. Now, one very cool thing we found here was that the color volumes they occupied were actually pretty similar, but so they were equally colorful along their distribution, but they didn't occupy the same color spaces. So they were equally colorful, but with different colors. Um, anyways, back to our question, our main question, right? What happens in island versus continental populations? And so what we did was compare the color volumes of males in the Galapagos or in San Cristobal and in the continent. And I know this graph is a bit of a spoonful, and as I explain it to you, you guys are going to be able to see the first big plot twist in our research. So on the y-axis on this graph, we have the color volumes for each individual, and on the x-axis, sorry, we have two split violin plots, which show the sorry, which show the one second. So it's a probability of density of the data along different values of the y-axis. Sorry. Along with a box plot that shows the mean value of gecko populations and the mean value of continental populations in green, and the mean value of Galapagos populations in orange, along with the interquartile ranges for the data. So as you can see, first big surprise, it turns out that continental geckos are more colorful than insular ones. And this was really surprising because colorfulness is a measure of conspicuousness. And if you remember, this is completely the opposite to what we expected. So the next thing we did was actually check different body parts or different, body colors from the geckos, right? So we compared adjacent colors with one another. And in order to do this, we used this unit called the just noticeable difference, which, as we applied it, lets you know how contrasting two colors are, or how different two colors are for the potential, for the eyes of a potential predator, for a bird predator in our case. And what we found, again, big surprise, is that continental geckos have more contrasting colors than insular ones, both for the yellow versus dark head patterns. As you can see, the average is way higher and for continental ones, but also for the blue spots versus dark sides that you see on the left of this graph. It turns out that they're more contrasting in the continent. Now, another thing that it's pretty interesting here is that the insular geckos have a wider variance in the distribution of just noticeable differences, and this was really surprising to us. One more thing that I need to mention in this graph is this dotted line in the one just noticeable difference value, which represents the threshold after which or before which the potential predators or the eyes of the potential predators cannot differentiate at all the two colors that we're contrasting here, right? So the next step was to compare colors to the actual environment, right? So far we've only talked about the geckos, but now we needed to talk about the geckos in their environment, or analyze the geckos in their environment. So in order to do this, we had sampled, we had small samples of the substrate from which, like from the spot where we had first spotted each one of our gecko individuals. And we took reflectance curves from them in order to compare them to the different colors in our geckos. And what we found, again, big surprise, for the yellow head, for the yellow head patterns, for the throat, and for the blue spots, Again, the continental geckos are more contrasting to their substrate than the insular ones. And this is particularly revealing, I think, in our data on throat and yellow heads, because the average in islands for both of those measurements, or for those both of those body parts, are below that threshold we spoke about. They're on average, they cannot be discerned by birds, the yellow and the substrate color. On the blue spots against the substrate, we see that the average, again, on the continent is a bit higher, and the variance in the insular populations is wider as well. So let's go back to our predictions for a second, right? Sorry, my throat is itching. So we expected this narrow conspicuousness distribution or variance in the continent, and we expected insular geckos, due to a reduction in predation pressure, to become a lot more contrasting, right, or a lot more conspicuous. And we actually tested for predation pressure, and our data suggests that they are under a lower predation pressure. However, our color contrast, or our conspicuousness data, follows a pattern closer to this doodle, right? They're not as conspicuous as continental geckos. And this sort of blew our minds, or my mind, for a long while, trying to look for an explanation for this. I turned back to the literature and found this research on island birds. So Duteland, back in 2016, published this study that compares conspicuousness of insular species with their closest continental relatives. And as you can see in this graph, both for males in blue and females in red, mainland populations have a higher color volume value, or as we call it, are more colorful than insular populations. So this same pattern was described for other groups, but we didn't find anything on reptiles so far. But still leaves us a huge doubt, like, why are island geckos less conspicuous? So one possibility is that just the founding population, the first geckos that got into the island just by chance, were less colorful or less conspicuous than the average continental populations, right? And the genetic pool in the island is just unable to produce more conspicuous phenotypes just by chance. And one of the reasons this is a hypothesis that's hard to test for us is because we are not sure from which continental population the insular population came from. And we actually attempted to answer this question using a couple molecular markers, but sadly our data was inconclusive, as you can see in this haplotype network, where that shows relationship between different or genetic relationships between different haplotypes. The continental population, sorry, the insular population sort of comes from all over the place. And if you break it down further, the same pattern is shown in the continent. Geckos appear to be moving all over the place, according to the few molecular markers that we used. There's definitely a lot of work to be done in this field, specifically in genetics, to better explain this phenomenon. Another reason why island geckos could be less conspicuous comes from a hypothesis that is put forward again by Duteland, which is a reduction on interspecific recognition, or the need for interspecific recognition. So what he finds in his study, or what he finds in his study, is that species that live in sympatry with more same family species tend to be more colorful, or have stronger signals. In this case, he measured the number of color patches they have in their body. So let me explain this graph real quick. The blue line represent males of different species, right? And in the x-axis, sorry, the number of sympatric species they live with, while on the y-axis you have the number of color patches they have. And for males, they find that if you live in sympatry with more same family species, the messages appear to be stronger, and it makes sense. It would be a bad idea for females to spend a lot of energy signaling to the wrong species, or even to avoid hybridization. It would make sense to be able to differentiate different species. Now, for our gecko, this actually is a pretty good explanation, because along its distribution in continental Ecuador, it lives in sympatry with several other species of the same family. But in the island, not only he's the only member of the family that's been introduced in San Cristobal, but it's the only diurnal gecko. So it's probably under a very low interspecific recognition pressure, if we may. Another potential explanation is reduced intersexual selection, right? An actual reduction on the sexual selection pressure we were talking about originally, and this could originate from different forces or different factors. Sorry. On one hand, on island populations, we would expect lower genetic diversity, right? Because of founder effects or bottlenecks, we'll expect island populations to have a lower genetic diversity. So it wouldn't make sense for females to spend a lot of energy choosing for the right male when the genetic makeup of most males are pretty similar. So this could turn out into a lower intersexual selection. Another interesting idea has to do with parasites, and this could also affect intersexual selection. So it's been shown in several studies that there's a relationship between parasitosis or even parasite resistance and sexual messaging, right, or sexual signaling. Sorry. So brighter males or more conspicuous males have been there's been found a relationship with a higher level of resistance to parasites or lower levels of parasitosis. And what we expect in islands, because diversity in general is lower, there could be a lower diversity of parasites as well, and this could also turn into a lower sexual selection. If everyone can message this resistance to parasites because there's a lack of them, the message is no longer honest, and it doesn't make a lot of sense to concentrate on that message from the female's perspective. We know there's several other reasons why this pattern could be happening. However, the time is becoming short, and so I would like to go straight to the take-home message for this presentation. So we found a very cool pattern. Although there is a lower, or there appears to be a lower predation pressure on islands, conspicuity is actually higher in the continent, and this was completely unexpected, as mentioned. So the thing that I want you guys to keep in mind is that there's still a lot of work to do in order to better understand the evolution of conspicuousness in general, and particularly in island populations. Additionally, I wanted to mention the huge opportunity that the introduced populations or introduced island populations present as a natural experiment, but also to let everyone know that they do come along with a bunch of complications as well. Finally, I wanted to thank the whole team that worked with me on this project. This was definitely a group effort from 4 different labs in 2 different countries. I want to thank my advisors that were always there with quick answers and really helpful. They have been very helpful. I want to thank everyone that was out there in the field with me, and everyone that was out in there in the lab with me, guiding me and helping me. The team has been amazing. I also want to thank the unofficial team, the whole army of people that helped me in different ways. The whole Evolvert lab, guys, thank you so much. You have been amazing since day one for in the development of this project and the whole experience. The whole army of people that helped me build the geckos, a bunch of volunteers, and from students to other people in the lab were always there giving me a hand. Everyone at the zoology lab in Quito, they helped me out with a bunch of ideas as well, with all the logistics of sampling all over the country and all over the islands as well, all over the continent. Everyone that helped me understand what the heck I was doing with R, with statistics, with developing the idea of the project. Everyone in this page had a very, very important role in this project. And finally, this slide makes me really happy because I cannot fit any more pictures, and this slide is dedicated to my family, both the ones that are genetically related to me and those that aren't, both in Bogota and in Quito. These people kept me sane throughout the whole process, and for the last 2 years, never complained over me constantly talking about geckos and colors and islands and evolution. Thank you so much, every single one of you. Muchísimas gracias a todos. Esta es para ustedes. Finally, I want to thank everyone that's here listening to the presentation, and the rest, I guess I'm ready for questions.
Okay, Mateo, thank you so much. I have to apologize because I completely forgot your co-director, who is Diego Cisneros, and I also had to thank him. So we will move on to questions from Devi. Thank you, Devi.
Hello.
Hello, Devi. Thank
Mateo,
you for
that was
being
a
with us.
wonderful talk.
Thank you.
And an amazing project. Your acknowledgement slides made me really happy too, and I don't even know the people in there.
I wish you did, man. Every single one of them are amazing.
That seemed like an enormous amount of work. It's great to see, a project with so much work on field work where you really get to understand your animal. I was particularly interested by the finding that you've got higher variance in the colourfulness, I guess, of the island populations, because higher variance kind of implies relaxed selection, whether that's relaxed sexual selection or relaxed selection due to species recognition. And at least for sexual selection, a very strong influence on the strength of selection is population density. Mm-hmm. And I was wondering, from all your field work, whether you could what your sense was in terms of the population density for island populations versus continental populations.
Sure. So we were joking around with some of the people that were in the field with me, specifically Leah, that in the islands, if you kick 10 rocks, you'll find 7 geckos. So where you could find them in the island, they were really, really abundant. Yeah, no doubt there. While in the continent, it's actually really interesting. The distribution of this gecko in the continent, there were whole areas where you could find none whatsoever. And then if you found one, all of a sudden, like in a small area with like a microclimate, microclimatic differences, you'll find a couple more, but nowhere near as densely as in the island. They were definitely more abundant there.
And so how do you think that might affect the strength of sexual selection or the opposing selection forces?
Well, regarding sexual selection, there's this idea that in higher densities, since males are finding each other all the time, perhaps spending the amount of energy to actually compete with one another is no longer advantageous, and so perhaps you'll expect a lower signaling strength because of this, because there's constant competition with one another. If the messages are directed to other males to try to keep territories or these kind of things.
Yeah, absolutely. I guess it could select for different kinds of strategies rather than relying on color signaling. And I guess related to that, what's, what kind of evidence is there for the role of color in signaling in geckos?
In general, I didn't, well, I guess in the few groups of diurnal geckos that we have here, there's some research done on like behavioral signaling, such as tail waving, and even showing the throat, and it appears to be in different groups. It appears to be both signaling towards other males, trying to keep like a territory in a way, or a small territory, and also towards females. I don't know if I answered your question or.
Yeah, I was just wondering, because I don't think there's been that much work done on color signaling in diurnal geckos.
Yeah, well, there aren't many diurnal geckos either. It's like a small branch of the the gecko diversity, and there's very little work on in general, like signaling in geckos. A little bit more on sounds, like the sound signaling, but very little on colors, because geckos are not usually the best example of colorfulness. A lot of the efforts that use like models that, such as birds, that are known to be colorful, or even frogs.
Even though there are spectacular diurnal geckos, like
Absolutely.
the Malagasy day geckos, they're amazing.
And I know I'm really how do you say this, sesgado. I'm really like, I have my mind set on one group right now. Gonatodes is amazing. Gonatodes caudiscutatus is just but an example of the huge variety of colorations these guys present, and not only colors, but the patterns that they have are totally amazing.
Well, that actually leads on to another question I had. You mentioned species recognition and the number of similar species in the community, and I was just wondering, on the mainland, do you get a lot of, or not a lot of, but you get a few similar species that are sympatric, so that is species recognition a real contender as an explanation in this system?
That's a really good question. There's still a lot of work to be done understanding the distribution of small reptiles in continental Ecuador, as the diversity, the mere diversity, has really make everyone like spread the focus on the work on small reptiles. But there are a couple, well, I know they live with other same family species, but I can't tell if actually you can sometimes find other geckos that are pretty similar, more closely to the females than to the males. However, there's a really exciting project like that I have been thinking about. This guy has also been introduced to the other side of the Andes, so towards the oriental side of the Andes, and there he does live with another species. He's sympatric with another species of gonatodes. So I'm kind of sort of excited to maybe take a tour and measure those colors and see what's going on over there.
Yeah, I remembered one thing that I thought of. You know how you measured geckos from the island and continental populations, and I think you had about, I don't know, 37 or 40 specimens from each. Do you have those specimens? Did you keep the specimens?
Not all of them. I did collect a small sample from each location I went to, so I have quite a few of them, but not all of them. Yeah.
The reason is that if, as you say, the island populations are much more dense, it could potentially select for different forms of competition or sexual selection. And so you might find, for example, that the island populations have larger testis size, where, you know, if it selects for more post-copulatory rather than pre-copulatory sexual selection. So you could compare different sexually selected traits, and testis size is an easy one that you can measure on specimens. That was just a thought.
That sounds really good. That sounds really exciting. Thank you very much.
Yeah. And one other quick question, I guess, related to, it's wonderful that you showed that predation pressure differed between island and mainland populations. So the big unknown is, I guess, the other selective forces, which is why I'm focusing a little bit on that. I'm mainly asking you to speculate. But do you know what we know about Gecko Vision?
Again, a field that's been studied very little. I know there's one like report on like the ocular system of gonatodes, and they actually are similar to bird eyes in the sense that they also have this like this extra I'm about to say cone. Yeah, this extra cone that we don't have. So we have the red, the green, the blue, and the violet one, and they too have it. But there's really not much more that has been done with this group, sadly.
That spot looked like it could be ultraviolet reflective. Do they have strong ultraviolet in that blue spot?
I expected a lot of like ultraviolet reflectance there, and I was surprised to find that there was a little, but really not that much. Yeah, that was one of the first things that like when I started reading about the topic, I was like, this guy's certainly reflecting ultraviolet, but
yeah,
not that much.
Yeah, it was interesting, the comment that you put up from John Endler to say what will look conspicuous to predators will look conspicuous to females. That's sometimes true and not always true. But I think in this case, you know, given what we know about lizard vision and bird vision, it would be true.
I hope so. Yeah, definitely.
Yeah.
I think it was back in the 80s, it wasn't as clear how different they actually can be, like the two observators. But yeah, it holds up, or I hope it holds up in this case.
And I guess the other thing that we're learning more about is the role of pattern, because these geckos have quite interesting reticulations, and that often creates some distant dependent effects. So, you know, in the photo, close up, the geckos look really colourful.
So
and high contrast, but when you move further away, the colours merge and they're actually really quite cryptic.
Yeah, and another thing, but going back to the antler thing, he measured size and number of spots, and actually we attempted to do this. We had like we took pictures from every angle for the geckos as well. Sadly, I'm not confident of what I found. We then compare these color patterns, but I'm not too confident on what I found, because it turns out having a gecko hold still in a straight line is a lot harder, and having a couple hundred of them do it is even like almost impossible. But something very interesting is that preserved geckos actually they lose the color, but the pattern is still noticeable in the difference of shades of white to dark. So I'm actually pretty excited about the idea of checking the patterns, both for the geckos I have collected and other Gonatodes caudiscutatus from museum specimens. And I think there might be something interesting there as well, to see if there's different phenotypes at work.
Yeah, as you say, so many more possibilities. I think my time's up. Again, congratulations, wonderful
Thank you so
project,
much, Devi.
great talk and thesis.
Thank you very much, and thank you for being here. It was really, really an honor.
Thank you, Devi, so much. So we will move into questions from Andrew Crawford. Thank you, Andrew.
Andrew, I think your microphone is muted. Sorry. There we go.
How about now?
Yep, perfect.
All right, very good. Wow, that was a great talk, Mateo.
Thanks.
I love hearing a story when you started with, I'm going to tell a story. It sounds like somehow like informal, non-professional, but I think it's the best. You hook somebody in and then bring them into the science. And it's so nice to have like a clear hypothesis and prediction, and then let's go. And then even more nicer, in my opinion, is when you get something you didn't expect. Because if you had said, oh, well, fewer predators are going to be more bright, and you got more bright, I said, look, okay, yeah, we thought that. So it's maybe harder for you to write up and present and argue, because now you're kind of like a
surprise,
looking for the explanation. But I don't know, I think we're going to learn a lot more that way.
Indeed.
Or at least I am.
You should have seen me the day I started, like, actually got these results. I was completely bummed out. I didn't realize how cool they were until I actually analyzed them. I was like, what? This makes no sense. I actually doubted the whole work,
Well, you would have been more bummed out if all your p-values were also really high. And then you'd have...
What is this?
But that was my comment on the written thing, is some of the p-values, but that's a boring topic for today, so we can talk about.
I actually would love to ask you a few questions after, like, at some point later, and some recommendations.
Sure, yeah, no, con mucho gusto.
Great.
So, and I had a Devi talk. I think she started getting into some of these same things I was going to ask. But I can follow up. So what about, I think you mentioned alternative responses. Let's say there's fewer predators. Let's say there's still just as much sexual selection on this trait. So what about behavioral responses in the two contexts? Do you think they're behaving?
Well,
different. Do you observe like anti-predator behaviors? Are they like the cormorants? You can just walk right up to them. Hi, Gecko, because there's no predators. Or
it's funny that you mentioned that. When we were first developing the whole project, we were wondering how to test for this predation pressure, because committing to building a bunch of paraffin models is it's a whole thing. So we actually considered measuring like distance, like I forget the name of the process, like when you approach the gecko and see when they react and when they run away. But we ended up not doing that. But I can definitely tell you that they were a lot easier to catch in the island. So I actually have a few numbers in my head. For every like 10 geckos you spotted in the island, we were able to catch 8, maybe 7. But when I started working in the continent, the first place I visited was the northern Choco forest, and I thought I was broken. All of a sudden, these geckos were disappearing completely. I spotted one on a rock. I approached, and it was nowhere to be found. So definitely, they were like quicker to had quicker responses, and they actually hit a lot better, I guess.
Yeah. Okay.
It's another good explanation. Yeah.
Are these geckos also territorial? I think Devi mentioned or asked about the male-male interactions. I mean, the hypothesis, when I listen to the talk or read it, I don't know about you, but I'm thinking it's about male-female or female choice, but it's also male-male as well, right?
Well,
are they defending territories, or do they just give up on territories in the island for some reason? Or is that, I mean, this is, again, like Devi said, this is your chance.
Yeah, completely speculative. So another pattern that I was obsessed in the field, taking notes of everything. And one of the things I have next to every specimen information is next to who he was, next to which other gecko, or under who was he sharing the rock with, or the rock assemblage, I guess. And we did find more, and this is completely me, like observational, right? We did find darker males with brighter heads usually shared rocks or had females closer by. And in some cases, we did find the same male in the same rock or the same few rocks in more than one occasion. So there's sadly no information on Gonatoisca biscutatus specifically if they are territorial. However, other members of the genus are, so one would expect them to be. And from this behavior in the island, they probably were, or they probably are.
Okay.
Yeah.
Okay. And you said that your gecko data from the continent was in a few different habitats, and you had that map, and you mentioned there's Esmeraldas, which is wet, and there's the south, which is dry. And maybe the island is a little more homogeneous, and you showed a picture of the island. Do you think, and I totally speculation on my part, what about shading? Because that island, what if it's really flat or they're more out in the open? And so if you're more in the shade, if you're more in forests on the continent, maybe you need to be more brighter because you're not getting them enough light. Is that a factor, do you think? Or
that is an interesting idea, and we sort of played with that around a little bit. But the thing is that these geckos we never spotted a gecko in direct sunlight.
Yeah, not even on the islands either.
Nothing. They're always.
Yeah, you did say that in your paper. Yeah.
Yeah, they're always like under the shade, and apparently they're really, really sensitive to temperature increases. So behaviorally, they spend all of the time either in shady areas, or if the sun is about to hit them, or if they're exposed to sun, they'll go under the rocks or hide. But yeah, we not once spotted a gecko in direct sunlight.
Okay. I was interrupted a little bit by some small children running around here, but I hope Debbie didn't already ask this question. But you mentioned parasites and other alternatives. What about, I didn't hear anyone mention diet. Did you mention diet? I know because birds get some of their colors from diet, as we know. And I don't know about your non-avian reptiles. Could that be a factor in?
Definitely. I mean, these guys are, some studies on other members of the genus have been conducted on stomach content, and they mostly eat invertebrates, right? So little invertebrates they can catch. Again, it's a big who knows, because there's almost nothing known of the composition of or how they actually produce the carotenoids for the I'm even assuming they are carotenoids for the yellow heads and whatever pigments they use for the blue patterns. There's very little known. However, it is a possibility that island geckos have something less.
Yeah. Well, in general, is there any evidence from any squamate where you change their diet and they become less colorful? I should know these things, but I'm too focused on frogs, I think.
A good question. I actually have not checked on that at all. I know there's several examples on birds, but
right, that's what made me think of the birds, right? Of course, yeah.
There's probably information on this. There's several geckos that are used as pets, and they tend to be colorful ones, such as the like leopard gecko. So it might be a good idea to check if like dietary compositions for these geckos incorporate some sort of source for a pigment. I'm gonna check on that. That's a really good idea.
Okay, and so in your manuscript, sorry, not to call it a paper, but I'm sure it will be a paper soon. It's
Thanks, man.
I
really appreciate it.
It's nice to see something so looks like a finalized thing. It's not. I don't know. I don't think there's anything left. If you don't want to add anything, it looks great. Just maybe something from the discussion or something, but it looks great.
Thank you so much, man. I appreciate it.
But so you said that let's see, I'm looking here. So you put out all the models, and then sometimes mammals come along and pick at them, and you throw those data out, and you're just looking for birds.
Yeah.
So just a crazy idea, probably not. But what if there were other non-avian predators on the islands, and then so maybe your lizards are not as released as we hoped? Not really.
I mean, it could be, but there's very little. Like there are very few mammals in the island. Actually, as far as I know, I might be saying something wrong here, but as far as I know, in the highlands, there's a couple of species of rodents, there's a big there's a very large population of cats that have been introduced there, but I never found any like bite marks from any other teeth pattern than from rodents. So it's unlikely, but again, it could be. Oh,
Okay.
sorry.
Yeah, Devi's jumping in there on the chat. That's great.
That's awesome.
Yeah.
I don't know if everyone else can see this. Maybe I'll read it out loud.
But you can read it if you want. Yeah.
Yeah, sorry. So, RE diet effects on color in lizards. Yes, there is some evidence, but lizards vary in whether the yellow-orange color is produced by carotenoids acquired by diet versus color pteridines self-synthesized. The blue is produced structurally by iridophores. That's very cool.
Okay.
Yeah.
All right. So it's not a crazy question.
Yeah, absolutely. And
okay.
It's a good.
So
sorry.
So I share Devi's observation that variance is really important. Equally, I mean, there's so often people are focused on comparing means or medians, but in this case, for evolution, I mean, evolution is is really acting on variation in populations, and you got that. So in various of your traits, as you mentioned and she mentioned, you get higher variance, which would suggest maybe a release, or be predicted by a release scenario.
Yeah.
And I don't know if you put a statistical value on that. I didn't see that. You just like observed it, but you could do that too. If I didn't, maybe it was there.
I think it's in the paper, but not in the presentation. Yeah, sorry.
Yeah. Okay. So I just want to throw out alternative explanations. Also, you had that one. What's it called? Your one haplotype network, and so the island is sampled from two sides of that haplotype network. So that would tell me either there was probably multiple arrivals or multiple invasions, or one big one that came from different places. And if those haplotypes came from different parts of mainland Ecuador, then maybe the alternative explanation that you might want to check before you put too much stock into it. What about the alternative explanation that you're getting multiple invasions of the island from multiple sources in Ecuador, and therefore you get a big variance? Whereas if you sample one place, but then you hold on, but then your data is sampled from, you're summing all over the Ecuador too.
Yeah.
Right. So okay, no, that wouldn't work then. I guess I answered my own question.
No. I actually went through that thought process already, and that's why I was following along. But and one big if in continental introduced populations, in insular introduced populations, is that we really don't have a clue if there are multiple entrances, and it's a huge possibility because they appear to be coming in by boat, and they're still bringing in produces to the island by boat since always. So maybe we're getting geckos all the time in there. So it's one of the big problems we have when working with these guys, or big things to keep in mind, I guess.
Yeah.
I think Andrew is breaking up. Or am I breaking up? Okay. So I think we're having technical difficulties with Andrew. Sorry about that. Thank you very much, Andrew, for all the comments and questions. I really appreciate it. And I'll be emailing you very soon with a bunch of additional questions.
Hi, Adri.
Hola. So thank you, Andrew, for all your questions.
Yes.
And it looks like we only have one question, so maybe Daniel can post it.
So should I read it out? Juan Manuel Guayasamín is asking, I had a similar question. Perhaps introduced cats and introduced rats might have a selective pressure on these geckos. Sure, it's definitely a possibility. From the models I collected, I never found any like evidence that like of bite marks from anything else other than in terms of mammals, other than rodents. But it's definitely a possibility, and since island populations, like introduced populations of cats and dogs are pretty large, they could definitely be changing or having a strong effect on the evolution of everything in the island, really. It could be, yeah. Thank you, Juan Manuel.
Okay. So we will continue with Diego Cisneros from Universidad San Francisco de Quito. Thank you, Diego. Maybe open your microphone.
Thank you very much. And first of all, congratulations, Mateo.
Diego,
felicitaciones. It
muchísimas
has been
gracias.
to work with you. And well, I've been along the process working with Daniel and you. But I had one question, and it's related to what Andrew already mentioned. How do you think that the extensive introductions and reintroductions of populations in the islands, and also the geographic expansions that are clear in the continent, could be affecting your results? And more importantly, how do you think that you can, if you could continue with this project, how would you try to see and measure and research that part?
Okay, so reintroductions into the island, I think, are a big source of noise in our results, or are probably a very big source of noise in our result if they're occurring. And this idea of the geckos expanding from expanding in the continent and moving around a lot is definitely another factor that makes the model not as simple to figure out. But I guess we do need to put a lot more effort into the genetic part of the research. Sadly, the approach we took, or the attempts we had with our genetic observations were pretty inconclusive. But I think this is because we only used 3 different markers, and definitely one of the goals for the near future is to use the genetic material we have to expand this, possibly, well, for more markers, or ideally for like full genotypes. That will be a very good goal, or a very good, cool thing to do, and to better understand what's truly going on with these guys.
Thank you, Mateo. Another thing that you didn't mention, probably because of the time, you found some morphological differences, especially in a few populations. Could you place that variable within the general context of what you have presented today?
Sure. So for everyone else, what Diego is referring to is specifically a couple populations in two locations in the dry forest in the southern part where we sampled, that were mini geckos. They were miniature geckos. Gonatesco discutatus is a very small gecko to begin with, but these guys were probably half the size, and this was a population thing. Both males and females, very drab colors, very sexually mature, were tiny little geckos. And this phenotypic variation got me thinking that there's probably some populations, and also where these guys were, they were like in the middle of a somewhat humid environment, completely surrounded by dry forest and not even dry forest, almost desertic lands around them. So there's probably some isolated populations in the continent that have been probably, who knows, maybe even more isolated than the island populations, that could probably give us some really cool information on these guys, if we figure out the genotypic variation in them, and also definitely the ecological, the morphological colors, etc. They're a really cool model to keep on working with these guys.
Thank you. Thinking about something that Devi Stuart-Fox mentioned, actually, in the future, you could think about also some geckos that occur in Madagascar, pretty far from here, but are also very colorful and kind of not the same, but kind of the equivalents of our geckos here. And also another thing that we didn't think about is probably miniaturization. Now that you are thinking about size of geckos, it's actually that these diurnal geckos are a lot smaller than nocturnal geckos.
Yeah, absolutely.
So it's just something to think about.
Definitely. There's I think we open up sort of a Pandora box of questions, which is a dream come true for me. And a lot of work to be done with this model.
Do you have any plans for the molecular part? I know that you have done some analysis now, but I think that you have some other dreams. Can you tell us about it?
Besides the molecular part?
No, with the molecular part.
Oof, I would really like to analyze these guys in a paint a clearer picture using either more markers or go full genotypes for these guys. Sadly, the budget is sort of our limitation, or has been our limitation so far. But yeah, that's definitely the next goal, I guess. I think that this will really remove a lot of the noise we're seeing currently with our results.
Okay. In the chat, actually, in the internal chat, Andrew actually asked about these Madagascaran geckos. And yes, I'm not sure either that if Phelsuma, which is the genus, are actually sexually dimorphic. But well, interesting to think about diurnal geckos that are also colorful.
Yeah, and if they are colorful and not dimorphic, where's this colorfulness coming from? That's a really interesting idea to check as well.
Yes. Thank you very, very much, Mateo.
Thank you, Diego. It's been a pleasure working with you. It truly has.
Thank you.
Thank you, Diego. So we will finish with Daniel, your director.
All right.
Hey.
Daniel.
Well, first, internet connection is kind of unstable, so I was losing you guys for a while. But hopefully this will work. Are you hearing me okay?
Yeah, perfectly.
Okay, good. Well, Crawford.
Sorry, Daniel,
it's great to have real experts.
Daniel, sorry, you're sort of breaking up.
Yeah.
Perhaps
maybe
if you remove the video.
Yeah, I'll do that. Can you hear me there?
Yep, it appears to be better.
Yeah. I was saying that. Yeah, well, I wanted to Devi and Andrew. It's been a pleasure to collaborate with you through.
Oh no, Daniel, sorry, we can't hear you anymore, or I can't hear you.
Yeah, this thing is jumping around. Back?
You're back. You're back. Daniel, can you hear us? I think we lost Daniel.
Can you hear me now, guys?
Yep, we can hear you now. You were gone there for a second, but now you're back.
Can you hear me now?
Yep, I think we have a little bit of delay, but we can hear you.
Okay, I'll just make it short. Just wanted to say, well, Mateo, I think you did an amazing job. Congratulations. Really exciting to see everything you accomplished. You that was great. I think that you did a job today with the talk. Your enthusiasm is contagious, and I learned a lot from this project with you. And you were it was happy. You're my first, I think. Yeah, you're the first international student to finish his degree with us, and it's been.
Oh no, Daniel, I think you're gone again. Daniel, I think you broke up again, and I was really enjoying what you were saying. It's a bad moment to break up.
Hey.
Daniel. Oh.
I think he's gone.
Okay, I think what he was trying to say is that he's very proud that you are his first international student to finish his master's degree with him. So congratulations.
Thank you so much. It's been
and I enjoyed your presentation a lot. Thank you so much. So I guess we will finish without Daniel. Let's try once again if he says something.
Sure, hopefully, yeah.
Daniel?
You guys hear what I said? Or?
Choppy. I think you should repeat all the nice thing you said.
Just write it in the chat and I'll put it, I'll say it to him.
Yeah, we'll talk later.
Okay.
Thank you so much. It's been a true honor working with you as well, Daniel. I don't know if you can hear me
thanks
clearly,
everybody. Ciao.
but it's been amazing. And for everyone else, thanks for being here. Adriana, thank you very much. All the advisors, thank you so much for your comments and your in general the conversation today. It's been amazing and really has given me a ton of ideas. And everyone watching, thanks.
I also want to thank everybody in your evaluation committee and Daniel, of course, for directing your thesis because this enriches all our graduate school. So thank you and congratulations.
Thank you. Thank you so much. Thanks to everyone.
Okay. Bye-bye.
Bye-bye.